Hey everybody, welcome back to General Biochemistry Lecture. We are now going over Chapter 8, only Sections 1 and 2, introducing more macromolecule players, nucleotides and nucleic acids. Here's the key principles for this chapter. Make sure that after you go through this lecture that you can read through this and kind of understand all of these different principles and be able to expand on them a little bit based on what I've introduced. But we'll start off with some basic definitions and some conventions. Every field has, their own language and their own conventions. And in this course, you're getting a crash course in a lot of different languages. You can think about it in that way. So first, nucleotides. Okay, they have a pretty varied role in metabolism. So you'll see nucleotides used as energy currency in metabolism. They also provide essential chemical links in response of cells to hormones and other stimuli. They're structural components of lots of different enzyme cofactors and metabolic intermediates. And most notably, they're the constituents of nucleic acids. So DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. We're going to be focusing on the last bullet point here with DNA and RNA in Chapter 8. We'll get to all the metabolism stuff soon. Let's talk about the functions of DNA. Really, the only thing that we know DNA does is it stores biological information, and it allows for the transmission of that information between generations. So it's a really stable way to store information about a biological specimen. A gene is just a segment of DNA that has the information required to make some kind of a functional biological product. That could be a protein, or that could simply be an RNA. I say simply, RNA is anything but simple. You'll see that. There's several classes of RNA. We've got ribosomal RNA, or rRNA, which you'll be responsible for knowing all of these little abbreviations for RNA. It'll just help you out a lot when you're reading. Ribosomal RNA is a component of the ribosome, and the ribosome is responsible for taking the messenger RNA and translating it into protein. Messenger RNA, mRNA, is the intermediate between DNA and protein. So messenger RNA is formed, it's also called a transcript, so an mRNA transcript, just because it is a transcript of DNA. That it has the code for making the protein. So it's going to bind to the ribosome and interact with tRNAs, the transfer RNAs. And these are adapter molecules that bind to amino acids. So it recognizes a 3-letter code on the mRNA, and it brings along with it the correct amino acid so that that polypeptide strand can be formed. So you've got the tRNA recognizing the mRNA, and then you've got on the other side, this growing polypeptide chain. We're going to cover all of this at the end of the class when we talk about, RNA and DNA metabolism. So don't worry about knowing that for now. These are probably things that you've already encountered in all of your biology, and we're just kind of refreshing at this point. The last one that you may not have heard a whole lot about in your biology classes is non-coding RNA. And these have a wide variety of functions. Everything from the spliceosome, which helps to splice messenger RNAs and make them into what they're supposed to be before they are translated into protein, to silencing different sections of the genome. So all kinds of things. Non-coding RNAs are really interesting. They have a wide variety of functions. Nucleotides and nucleic acids have characteristic bases and pentoses. Pentoses Throwback to chapter 7. So all of this builds, all the vocabulary when we're talking about sugars, some of that's going to apply to our conversation about nucleic acids and nucleotides. So nucleotides have three components. There's the nitrogenous base, there's the pentose, so the sugar, and then you have at least one phosphate. Okay, so nucleotide has phosphates. That nitrogenous base is either a pyrimidine or a purine. Pyrimidines are, I think about it as this way because you'll need to know the difference. Pyrimidine, long name, little molecule. So it's like opposites. The purine actually has the two-ring structure. And we'll talk more about this specific nucleotides and nucleosides in a second. Nucleoside, we're talking about the nitrogenous base plus that pentose sugar, no phosphate. So know the difference between nucleotide and nucleoside. You'll also want to pay attention as we talk more about nucleotides and nucleosides to this 2' position here. This is the determining factor for whether you're looking at DNA or RNA, if you're looking at deoxy versus just ribose, okay? These major purines and pyrimidines, you'll need to know these. Recognize them in their nucleotide state and nucleoside state. A, G, C, and T, those are all in DNA. Thymine is only in DNA. That's the T. Uracil replaces thymine in RNA. So you'll need to know which ones are in which molecule. And the answer is everything is in DNA and RNA except for thymine and uracil. So thymine's DNA, uracil is RNA. You'll need to be able to recreate these purines and pyrimidines. So be able to draw them. I'm not going to make you responsible for drawing nucleotides and nucleosides, but I do want you to be able to draw the purine or the pyrimidine. This table gives you some help with nomenclature. So if we're talking about purines, we've got adenine and guanine. The nucleoside for RNA is adenosine. For DNA, it's deoxyadenosine. So that 2' hydroxyl group is gone. There's no hydroxyl group there. Same goes for the comparison of RNA and DNA for all of these. The exception being looking at thymine and uracil. So since they only exist in DNA or RNA, we don't have to worry about those distinctions. So there's two different kinds of pentoses, which we alluded to. They're both ribose, but we can either have deoxy D-ribose, like in DNA, that's the D part, deoxy, or just plain old D-ribose, which is in RNA. They're both furanoses, closed 5-member ring. But again, the presence or absence of that hydroxyl group determines whether or not you're looking at a nucleotide that is for DNA or for RNA. This, as it's pictured, is for RNA. Deoxyribonucleotides, these are the structural units of DNA. There's lots of different terms that you can use, so I want you to be familiar with them all. It's kind of like learning the English language or any language where you have synonyms for things. This slide is going to be really helpful for recognizing the structure and getting to know the terminology for all of the deoxyribonucleotides. Then we have the same thing for the ribonucleotides, and these are for RNA. Notice that really the only difference is the presence of this hydroxyl group, and also we're swapping uridine in for thymidine. There's also some minor purine and pyrimidine bases, and pretty much it's the methylated forms. You see methylation a lot in DNA. You don't need to memorize these, but just know that they exist. You can also have nucleotides with phosphate groups at different positions. You don't need to memorize the different positions, but again, just be aware that this is a thing and they have a purpose. What you'll definitely see when it comes to signaling is cyclic AMP and cyclic GMP. So those are definitely two important ones to take note of, not because you need to know the structure, but just because it may crop up again in one of our case studies when we're looking at things that have to do with nucleotides. Now we've got all of these building blocks, but we have to have a way of stringing them together, otherwise we won't have a nucleic acid. The phosphodiester bond is what links all of the nucleotides in your DNA and your RNA. It's a covalent bond that joins all the nucleotides together. So what happens is you have the 5' end of DNA or RNA. So we're looking at this area. It is going to link up with the phosphate of the 3' of the previous nucleotide. There's going to be a covalent bond made, and I'm not going to require you to know the chemistry and draw the arrows for how the phosphodiester bond is formed, but just know that this is what it's called. It's a phosphodiester bond, and it's between the 5' phosphate on one nucleotide and the 3' hydroxyl of the next one to be added. So you definitely want to know that. DNA and RNA can actually be hydrolyzed under alkaline conditions. DNA is not as rapidly hydrolyzed, but RNA absolutely is. And the reason for this difference is because of this 2' hydroxyl. If you have a base that can steal away a hydrogen from that free hydroxyl group, then you suddenly have electrons that can do some work. And just like, let me erase that because I'm completely covering that P in the phosphate. Whoops. Just having all types of issues here. Okay. Let's try that again. These electrons can attack that phosphate because it's got a partial positive charge. Just like a carbonyl carbon has a partial positive charge, that phosphorus in that phosphate molecule has a partial positive charge. So it is ripe for chemistry when it comes to hydrolysis and you have a good nucleophile. So once you do that, then you can kind of break it apart. So you have, either a break in an RNA strand or something like that. There's some more terminology, and you'll see a lot of similarities when it comes to talking about DNA and RNA, so nucleic acid structure and protein structure. Be thinking about the similarities in terms of the terminology, because as we build up, we're eventually going to have a final. And that final is going to be taking all of these concepts and linking them together. I'll do my best to point out the areas that you should be looking at and trying to forge connections between. But you'll need to do that on your own too. So come to me with questions about how to do that if you need help. But you can think about biochemistry as, all of these different players. We've got all these different teams in the body. We've got team protein, team carbohydrate, team lipid, which we're going to be introduced to next week. And all of these things are coming together, and they're just having it's like a free-for-all fun day. You got kickball, you got dodgeball, three-legged races, all kinds of things, and they all have to interact with each other. But there's certain rules for how and when they interact. We're learning the terminology so that we can look more closely at those interactions and how all those interactions together form a functional human being or other, species like some animal or something, right? But we're focused on people. Let's just be honest. Medical field, we're focused on people here. Okay. So that was a brief aside to try to help bring together why we're looking at all of these things. It's so easy to get stuck in the terms and all the things you have to memorize and forget the bigger, broader picture of learning the players so you can see how they work together. All right. So speaking of learning players, some more terminology. We've seen oligo and poly several times now, so you probably already have an idea of what we're looking at here. An oligonucleotide is a short nucleic acid, so we're talking like less than 50 nucleotides. Polynucleotide, much longer nucleic acid. So the properties of the bases affect the three-dimensional structure of nucleic acids. All of the bases are weakly basic compounds. They're aromatic, and most of the bonds in these rings have some partial double bond character. So that makes them pretty rigid and fairly planar. We're not going to talk about, the small differences between the purines and pyrimidines in terms of, the purines have, are planar, the pyrimidines, maybe they pucker a little bit. We're not talking about that. But you do want to be familiar with these basic characteristics of being weakly basic, aromatic, and the double bond character. All nucleotides absorb UV light, and they have pretty strong absorption around 260 nanometers. And this is key in the lab. This is actually used to calculate concentrations of nucleic acids in solution. There are instruments called, I know that the proprietary name for the one that I'm thinking of is a nanodrop, but there are these little machines that literally fit on a table or a desk next to a computer that you can put 1 or 2 microliters of your nucleic acid solution on and put the little lid down, and you can look at the absorption and it will calculate the concentration of the solution based on its 260 nanometer absorbance and the molar extinction coefficient. If we're looking at nucleotides, they're actually pretty hydrophobic, and so they're not really that soluble in water at pH 7. But if you have more acidic or more alkaline conditions, then it's a little bit more soluble. But these things definitely don't want to be interacting with water at pH 7, and that's what we have a lot of in us, and we're around pH 7. So what do? Well, there's lots of stacking and base pairing. Base pairing fulfills the requirement of keeping all of the hydrophobic stuff together and sequestered away while keeping all of the hydrophilic stuff on the outside. So Watson and Crick, I'm sure you've heard those names several times in biology, and they were really instrumental in identifying the 3D structure of DNA. Base pairing is just the hydrogen bonding pattern between the complementary strands of nucleic acids. So typically, you've got A bonding to T and DNA. It can also bind to U if we're talking about RNA, and then G and C pair together as well. What you can see in this blowout here is the interactions between A and T and G and C. When I talk about you knowing the structure of the bases and being able to draw it, I also want you to know where the hydrogen bonding happens. So we're going to talk about that specifically on a separate slide, but I'm just pointing it out now that that's something you'll need to know as well. But notice how we've got all of this hydrophobic stuff in the center, and then what you have on the outside, so this part, is your sugar phosphate backbone. We talked about sugars in chapter 7. They need water to be soluble because they have all those hydroxyl groups that need some hydrogen bonding to keep them happy. And then we've got phosphate groups, this long phosphodiester chain, highly negative. So those are ripe for hydrogen bonding. So it's just like a protein when you think about the 3D structure and the idea of sequestering away the hydrophobic region, aka the nucleotides, away from, so I guess we'll call these the bases, right? Because the whole thing is a nucleotide. So the bases, the purines, the pyrimidines, they're facing each other and doing some hydrogen bonding, but they're also able to do some stacking, which is another way of saying van der Waals. And then you've got the sugar phosphate backbone that is incredibly hydrophilic and needs water to stabilize it. So now we're going to talk about nucleic acid structure in more detail. I was already getting to that because I'm getting so excited. Nucleic acids is another big interest of mine. Just like with proteins, nucleic acids have hierarchical levels of structure. We've got primary structure, and that includes the nucleotide sequence and the covalent structure, right? Then we've got secondary structure, which is regular stable structure that's taken up by pretty much all nucleotides, okay? Then the tertiary structure, we're talking about the folding of large things like chromosomes or tRNA, rRNA. Those things have, lots of different structural features in them. This is the Watson-Crick model for the structure of DNA. When you have two strands coming together, wound around each other, you actually create what's called a major groove and a minor groove. And these grooves are really important when it comes to protein binding and enzyme binding, because they are specific for, some bind major grooves, some bind minor grooves. So this is actually an important distinction. When you look at the ball and stick model here in the center, I think that the major and minor grooves are much more visible. So we've got a major groove here. That's this space here. That is much bigger than our minor groove, which is here. I remember being in grad school and having to look at a structure and identify major and minor grooves, and it was such a pain. It was one of those things that took me a little bit to really understand. And it was just so frustrating to be like, okay, I'm supposed to just look at this structure and know what a major groove is and a minor groove is just based on this. Oh my gosh. It was one of those like, I'm ready to tear my hair out and eat it type of moments. But I got there. I finally got it. Anyway, in addition to the major grooves and the minor grooves that we create, we also have to talk more specifically about the hydrogen bonding between G and C and A and T. Between G and C, you're going to have 3 hydrogen bonds. Between A and T, there's going to be 2. This is the kind of traditional bonding. Okay. There's also something in your book that talks about Hoogsteen pairing. We're not talking about Hoogsteen. Okay. Hoogsteen is another one of those things that plagued me initially when I was in grad school, but I finally understand it. But Hoogsteen base pairing, it absolutely is relevant, and it's something that needs to be considered if you're interested in DNA and RNA and the proteins and enzymes that bind them and do chemistry with them. However, for our purposes, I'm not going to make you understand Hoogsteen pairing, but it's just an alternate way of the bases interacting, so it's different hydrogen bonding that causes for differences in structure that can be recognized by different proteins. Okay, so there's that. More descriptions about DNA. The strands of DNA are anti-parallel. So there's two strands, right? It's a double helix. I can't believe that's the first time that I'm saying that. I think it's one of those things that everybody knows that, right? It's a double helix. Anyway, that should be stated. DNA is a double helix. And the strands are anti-parallel. So what that means is we've got these two different strands. So I'll kind of color. There's one strand. I'm not going to color the whole thing. And we'll say this is the 3' end. And we get all the way down here. This is the 5' end. The second strand all the way down here. The 5' is actually up top with the 3' of the other. So these are anti-parallel. The phosphodiester bonds are running in opposite directions. It's going to be really important to understand that when we're looking at DNA and RNA metabolism, because understanding the anti-parallel nature of double-stranded nucleic acids will help you understand the mechanism for how they how we replicate and repair these molecules. So it goes without saying that parallel means you would have 3' and 5' running in the same direction. The anti-parallel chains are complementary. So that's how we get our base pairing. If you see an A in one train, you're going to find a T in the other, G in one, C in the other. Surprisingly, the hydrogen bonding between the bases doesn't contribute significantly to the stability of the DNA. I know. Mind blowing. It doesn't contribute to it, then what does? What actually helps the most is there's a ton of metal that is protecting the negative charge of that phosphate backbone. So the outside, I'm going to go back just for a second here. So this part, the circles and the pentoses, this is known as the phosphate backbone. You may also see it called the phosphate sugar backbone. They all mean the same thing. We're talking about the phosphates and the pentoses of all of the different nucleotides, and that's on the outside of the double helix. All that negative charge from the phosphates has to be covered, okay? And you cover that with metal. When you're dealing with DNA or RNA, you're typically going to see a lot of magnesium ions. So that's Mg2+. And those ions will interact with the backbone to shield all of that negative charge. That helps to stabilize the structure of DNA. The other thing that helps with stabilizing the structure still has to do with the nucleotides, but it's not the hydrogen bonding between them. It's base stacking interactions. So when you stack the base pairs, there's van der Waals happening between each layer. So if we were to represent all the pairs, the base pairs like that, they're stacking in between. So this is the van der Waals. Which is also known as base stacking here interactions. Base stacking interactions is specific to nucleic acids. It falls under van der Waals, just to try to help you keep all the terminology straight. All of those stacking interactions stabilize the double helix of the DNA. If you have more GC context, then you have a more stable molecule. And this comes into play if you're doing work with nucleic acids. Oftentimes you will need to purchase or make different oligonucleotides for cloning and all kinds of purposes. And if you have a lot of GC pairs, it can actually be a little bit harder to work with because it's super stable, and sometimes they actually form, different structures that you don't really want. So it's something that you have to keep in mind experimentally when you're trying to make or purchase oligonucleotides. That's completely an aside. If you're interested in that kind of thing, we can talk about it all day long. So we already know that DNA has to be replicated. These are just the very, very basic steps of DNA replication. Again, we're going to get into that when we look at DNA and RNA metabolism towards the end of the course. But the first step in replication of DNA is you have to separate the parent strands. So in its current state, it's very stable. The information is not accessible. You separate those strands, and each of them serves as a template for creating 2 new daughter strands. So each new molecule of DNA has 1 parent strand and 1 daughter strand. The daughter strand is the one that is newly synthesized, that's complementary to the parent. DNA can occur in different 3D forms. So those structural variations reflect the different conformations of that deoxyribose. So remember that sugar, there are some areas where it can turn. The bonds are free to rotate. And then there's also rotation in terms of about the phosphate backbone, the phosphate sugar backbone. The 3 forms of DNA, we've got the B form, which is the most stable. That's the Watson-Crick structure. It's what you see everywhere. And that is relevant for us because, that's what you see under physiological conditions. You can also get the A form of DNA, and that's a right-handed double helix that has a wider turn. And I'll show you some pictures of this in just a second. You don't need to know how many base pairs per turn, but you should know the difference between B form and A form. And A form is favored in solutions that don't have water. So think about organic solvents and things like that. Then there's the Z form, which is totally, it's doing its own thing. It's a left-handed helix. And it's, got a zigzaggy appearance. And it's kind of elongated and slender in comparison to the other forms. So here we go. A form, B form, Z form. You've got a table on the right-hand side. You don't need to know all the numbers like the diameter of the molecule and how many base pairs per turn and stuff like that. But you will need to be able to compare A form, B form, and Z form in terms of, right-handed versus left-handed. And A form, you're going to see that more so in, organic solvents. You're not going to see it really in water. B form is physiological. It's the Watson-Crick structure. Z form Just know that it's left-handed and it's got kind of a zigzaggy appearance with that phosphate sugar backbone, and it's more slender. There's some DNA sequences that adopt some strange structures. One particular region, we're looking at palindromes. So a palindrome is something that is the same whether it's read forward or backward. We have words that are like that. It's the same thing in DNA. And in DNA, what we've come up with experimentally is, and some of these are just found in nature, in bacteria for sure, is these things called restriction enzymes, or we perform restriction digests. That means chewing up specific pieces of DNA or RNA or making a cut. Okay. They usually recognize palindromes. So you'll get an enzyme binding here, and you'll also get an enzyme binding here. But these particular places are where you can see some different structures in the DNA. You can also see a mirror repeat. So that's when you have a sequence, and then that sequence is like inverted and repeated. So if you had a mirror right here, then you would see that mirror repeat. Some of the structures that you can see, we'll talk about hairpins and cruciforms. So a hairpin can form when you've got two complementary pieces of nucleic acid that are separated by a short stretch. They can see each other and say, oh, hey, we can base pair. So you get this little circular piece at the top where there's no base pairing. But then you have this complementary section here that's holding it together. That's a hairpin. And it kind of looks like a bobby pin. The next form that you can have is a cruciform. And that's essentially like a hairpin gone crazy. So we've got two hairpins that come together because we've got all this complementarity. So we've got a double helix, right, that's already got its own base pairing going on. And when you've got these two regions of complementarity so close to each other, and then you've got just kind of normal sequence here, then it's going to end up forming a cruciform where you have these two regions of complementarity having some base pairing, and then you've got no base pairing going on at the top. Let's talk more about RNA, because RNA is really where you see a lot of these crazy structures. mRNA, the messenger RNA, that is what helps to carry the genetic information from DNA to the ribosome. Transcription is what that process is called, and we'll talk about that later on in the course. When you form mRNA, you can have monocistronic or polycistronic. Monocistronic means that the mRNA codes for one polypeptide. So one mRNA equals one polypeptide. But you can also see polycistronic, and that means that you've got one mRNA that leads to two or more different polypeptides. You see that a lot in bacteria, where you have one super long mRNA transcript and then you actually end up making like 10 proteins. mRNA is always single-stranded. Okay, it's got some other business to do with the ribosome, and it needs to be available to interact with the ribosome and those tRNAs that are bringing in the amino acids. So it doesn't have time for another strand. Okay, it's got enough relationships. It doesn't need the relationship of another RNA strand too. It forms its own right-handed helix, and the base stacking interactions are what stabilize this structure. It's strongest between two purines. So remember, the purines are the ones that have the double ring structure. So when you have long stretches of purines, that helps with the stability of the molecule. mRNA can base pair with complementary regions of DNA or RNA. And when it does that, it is anti-parallel, just like with DNA, the two DNA strands. Secondary structure of RNA can get pretty wild. The structure of two strands of RNA that are complementary is actually the A form. Okay, so DNA, B form, RNA, A form, right-handed double helix. There can be breaks in the structure that are caused by mismatches or unmatched bases, and that can result in bulges and loops and all kinds of things. You got internal loops between palindromic sequences, so you don't have to necessarily know all of these things on the slide. Just know that secondary structure of RNA can be pretty wild. There's a lot more going on than with DNA. DNA, you've got your double helix, B form, that's it, all day, every day. RNA, much more complex. You can have some base pairing in helical sections in RNAs, especially like tRNA or non-coding RNA. So what's shown here is a tRNA. This is a topology diagram. Just like with proteins, it shows the secondary structure. And these tRNAs are pretty huge. So they have different domains, the C domain, the S domain. You don't need to know anything about tRNA specifically, but there are specific domains within the tRNA molecule. There's a place where the amino acid binds, so there's chemistry there that allows for the attaching of that amino acid to the tRNA. Hairpins are really, really common. It's the most common type of secondary structure when we're talking about RNA. If you look at the B part of the figure, this is a very complex molecule. The folding, I think it's beautiful. tRNAs are very artistic. And I think that studying all of these molecules is really cool. So that's me geeking out about nucleic acids and nucleotides for about an hour. Hopefully there's something in there for you. We'll talk about this stuff in class. We'll do practice problems, all the normal stuff. But really what this is for is to give you an idea of what things look like, the terminology, so that when we move on to metabolism, we're not having to introduce vocabulary. You're pulling from what you already know, and we're applying it to all these different systems. So thanks for watching. Make sure that you participate and do the practice problems. And as always, if you have questions, send me an email or talk to me in class, after class, all those things. That's it for now. Stay safe.